Hello students, today we'll start a new topic, pharmacodynamics. In this particular session, I'll give an introduction of what is pharmacology, what is pharmacodynamics and pharmacokinetics, the various type of drugs and drug receptors, their types and interaction. The word pharmacology is derived from the Greek word pharmakon, meaning active principle or drug, and logos means study. Pharmacology is the study of substances, basically chemicals, as to how they interact with the living system, alter the regulatory molecules to activate or inhibit normal body processes. There are several branches of pharmacology like pharmacokinetics, pharmacodynamics, pharmacotherapeutics, chemotherapy, molecular pharmacology, toxicology, etc. Pharmacodynamics. It is the study of action of the chemical on the organism. Basically, what the drug does to the body. It includes the relationship between the drug concentration and the molecular, biochemical, and physiological effect of the drug on the cellular system of the body. While pharmacokinetics deals with the absorption, distribution, and elimination of the drug from the body. In short, what the body does to the drug. The study or branch of pharmacology that deals with as to how the substance or chemical is used to prevent, treat, or diagnose the disease is called medical pharmacology. While the branch of pharmacology that deals with the undesirable or adverse effect of chemicals on living system is called toxicology. The figure here illustrates the two major areas of pharmacology, medical and toxicology. The relationship between dose and effect can be separated into two components, pharmacokinetics, that is dose and concentration, and pharmacodynamics, that is concentration and effect. The three primary process of pharmacokinetics is absorption, distribution, and elimination. The concentration of the drug is the link between pharmacokinetics and pharmacodynamics, and it focuses on the dosage for the physiological effect. What are drugs? These are substances which are used for diagnosis, relief, treatment, cure, or prevent a disease and affect the functioning of the body. They are broadly classified into two groups, prescription drugs and non-prescription drugs. The prescription drugs are the drugs that are consumed under medical supervision. Non-prescription drugs are the ones that are sold over the counter, example aspirin. Besides these, there are some dietary supplements like vitamins, amino acids. These do not come under the prescribed drugs. The drug has three names. Chemical name, that is the molecular or atomic structure of the drug. Generic name, it gives the information about which group or class the drug belongs to. Example, antibiotic, beta blocker, etc. Then the brand name which is given or chosen by the pharmaceutical company that has patented the drug. After the expiry of the pharmaceutical company's patent, the drug can be sold under the generic name. Example, ibuprofen or the brand name Advil or Brufen. This is a non-steroidal anti-inflammatory drug to reduce pain and fever. The other common example is aspirin. Aspirin is a generic name. The chemical name is acetyl salicylic acid and the brand name is Ecosporin. The drug action could be stimulation, depression, irritation, replacement, cytotoxic action, antimicrobial or antibiotic action, or modification of immune status. The drug acts as a signal which is perceived or recognized by the receptor. The receptor are molecules that are present on the surface or inside the cell and specifically recognizes certain endogenous compounds. The drug receptor complex initiates a series of reactions which leads to a intracellular response. The secondary messengers or effectors act as signaling molecules to bring about the response. The first step is binding of the drug to the receptor. The strength at which the drug forms a complex with the receptor is called affinity. It depends upon the size and shape of the drug. The drug is smaller in size than the receptor molecule. The other factors are the number of receptors on the cell surface, the interaction between the drug and the receptor, the stereochemistry of the drug receptor binding site, and the force between or the intermolecular force between the drug and the receptor binding site. The cell surface has many receptors. However, the cell will respond only to some drugs or a spectrum of drug due to its affinity to the drug molecule. Higher the affinity of a particular drug to only one receptor, the action or response would be very specific and selective, with less side effects. The efficacy of the drug too would be high, that is, at lower dose gives potential response. Receptors are mostly proteins. They could be enzymes, membrane proteins, nucleic acids, and complex polysaccharides. The two major functions of a receptor is binding to the molecule or the drug and signal transduction on interaction with the molecule to give a response. Hence, a receptor has two domains, the receptor ligand binding domain and the effector domain. The characteristics of the receptor that makes it effective is its specificity, selectivity of response, and sensitivity. Interactions of the receptors with the ligands involves the formation of chemical bonds like covalent, electrostatic, and hydrogen bonds, or weak interactions involving van der Waals bonds. These bonds are necessary in determining the selectivity of the receptor. The size, shape, and charge of the drug determines the type of interaction and action. These interactions exhibit a high degree of specificity. The receptors on the cell surface are embedded in the plasma membrane and are hydrophobic. The intercellular receptors are located in cytoplasm or nucleus and bind to small hydrophobic ligands like steroids. Two models are proposed for binding of ligand to the receptors: the lock and key model and the induced fit model or hypothesis. According to lock and key hypothesis, a drug must correctly fit into the binding domain of the receptor to give the biological action. Like every lock has a precise key. This phenomenon is responsible for the specificity of the target drug receptor. In case of induced fit hypothesis, the receptor can change its shape to properly align to the target drug molecule to induce cellular response. In this model, the active site of the receptor is not static. The induced fit hypothesis is now replaced the lock and key hypothesis. The drug-receptor interaction regulates cell function by altering enzyme activity, permeability to ions, conformational changes, and genetic material. The major receptor families are transmembrane ligand-gated ion channels, transmembrane G-protein coupled receptors, transmembrane enzyme-linked receptors, and intracellular receptors. Ion channel receptors. These are transmembrane receptors. The binding of the ligand to the receptor brings about conformational changes and open up the otherwise closed ion channels. This leads to movement of ions like sodium, chlorine, calcium, potassium across the cell membrane, creating a gradient across the cell membrane. This leads to depolarization of the surrounding membrane and trigger the signaling pathway. The figure on the right represents a ion channel receptor. Examples to this are neurotransmitters, nicotinic acetylcholine receptors, GABA receptors, glycine receptors. Ion channel receptors are made of multiple peptide units. Example, alpha, beta, delta, gamma, and each of these contain 4 membrane-spanning domains. Binding of the ligand, example, acetylcholine, to these domain bring about conformational changes leading to the opening of the ion pores. This causes depolarization of the surrounding membrane, but its duration is very less, just milliseconds, even if depolarization is maintained. as shown in the adjacent figure. Here, the effector are, are the ion channels and the interaction between the receptor and the ligand is direct. Next, G protein coupled receptors. This consists of the largest family of transmembrane receptors. It is a heptahelical transmembrane receptor. Please see the figure on the page, with the N-terminal domain being extracellular and C-terminal being intracellular. The G protein floats on the membrane with the exposed domain in the cytosol. The third cytoplasmic loop of the transmembrane couples with the G protein, as shown in the figure. Binding of the ligand on the extracellular domain leads to coupling of the G protein with the cytosol segment. This leads to activation of signaling pathway. The effector here is an ion channel or an enzyme. The mechanism of activation of G protein coupled receptors. Here, in inactive state, GDP is bound to the exposed domain. The binding of the ligand to the receptor activates receptor and GDP is replaced by GTP. The alpha subunit carrying GTP dissociates from the beta and alpha subunit and activates or inhibits the effector, as shown in the figure below and in the next slide. The figure here explains the mechanism of action of G protein coupled receptor. The figure on the right explains G protein coupled receptor, how it controls the ion channel without secondary messenger. In case of G protein coupled receptor, the duration of action would be in seconds. Transmembrane enzyme linked receptors. In this case, the receptor consists of a protein that may form a dimer or a multi-subunit complex. The receptor here has two domains. One facing the extracellular surface of the cell where the binding of the ligand occurs, and the second the intracellular catalytical domain, which is an enzyme like kinases. On activation, the receptor undergoes conformational changes and affects cellular signal transduction by phosphorylation of different target molecules. The figure on the right shows an enzyme-linked receptor activated by insulin molecule, leading to autophosphorylation of the receptor. This initiates a cascade of signaling pathways. Examples of transmembrane enzyme linked receptors are insulin receptors, growth factor receptors, cytokines. The example below shows activation of enzyme linked receptor by growth factor. Binding of the growth factor molecule to the receptor activates tyrosine kinase domain. This leads to conformational changes and dimerization. This further leads to autophosphorylation of tyrosine, which in turn binds to the SH2 domain of the growth factor receptor bound protein 2. The phosphorylation of GRB2 activates a cascade of kinase action, leading to activation of transcriptional factors and finally gene expression. The fourth category of receptors are intracellular receptors. These receptors are intracellular in nature and hence the ligand needs to be a lipid-soluble one to enter the membrane and bind to the receptor. There are two main categories of intracellular receptors based upon their location: in cytoplasm and in nucleus. The ligand receptor binding activates the complex by dissociating the receptor from a variety of binding proteins. The activated ligand receptor complex translocates to the nucleus, wherein it dimerizes before binding to the transcriptional factors to regulate gene expression. The duration of activation and response could be hours to days. Examples for this are steroidal receptors. This figure summarizes the different types of receptors that we have studied. After the activation of ligand receptor complex, the signal transduction pathway initiates the response to pass the signal to the primary target. The main feature of signal transduction pathway is to amplify the signal originating at the ligand receptor complex and to protect the cell from excessive stimulation. This is done because repeated or continuous administration of the drug may damage the cell, like high concentration of calcium initiates cell death. To control this, the receptor becomes desensitized. Thank you.